I'm Dr. Joe Graham. Um, so Joe did his PhD and postdoc at Arizona State University, and there he studied spinal cord motor neurons, and he also started working on this tool to analyze neuron morphologies and generate artificial morphologies. So he generated these morphologies in a way that they cannot be distinguished from real ones, which is something really useful. He then continued with a postdoc at the Bluebrain project in Lausanne in Switzerland, where he did more work on this field. He extended this tool and he generated artificial neocortical neurons using this tool, which is really state of the art. So if you compare, he showed us his results compared to other tools in the field that do these artificial morphologies, and it really performs better than any of the other ones. So he's working on writing a paper and making this tool available. More recently, he's worked as a contract scientist at SUNY Downstate and Bill Litton's lab for 3 years. He worked remotely from Phoenix, and this is where we met and we became good friends. So here he was developing models of cortical circuits and also looking at dendritic function. And I'm happy to say that since February, he moved to New York and he joined my lab as a research scientist. And he's working on this NIH grant to develop a tool for modeling brain circuits. So today he's going to talk about one of his passions, which has nothing to do with the previous stuff, which is the evolution of the brain, which I think is really fascinating. And I really believe it will be very influential one day. So Joe, please.
Thank you very much, Salvador. So yeah, a few years ago, well, ever since graduate school, I've been interested in how nervous systems evolved. When I first really got evolution, it clicked. And all of a sudden, the world made so much more sense. Everything fell into place. And I've never gotten that from our understanding of how our brains evolved. And so I wanted to start looking into it. And I spent several years doing it. And I think I've discovered some interesting things. And I'd like to share them with you today. So yeah, I call this overall concept the adaptable gut hypothesis. Okay. So yeah, if you're looking into the origin of brains and nervous systems, the first thing you probably should realize is that we vertebrates don't have just one nervous system. People say the nervous system as if the brain and spinal cord were all we had. But no, we've got the central nervous system, the brain and spinal cord, peripheral nervous system in the body, sort of the somatic nervous system. We've got the autonomic nervous system, so the sympathetic nervous system, which sort of sits on the back wall of your trunk, and the parasympathetic nervous system, which is craniosacral. Yeah, so there's several nervous systems. So you can't just talk about the evolution of the nervous system. You should really look at all of them. So this is a textbook. I got this from a sort of high school / early college level biology textbook. And this is the nervous systems, except they're forgetting an enormous, very important one, the enteric nervous system. The gut gets no respect. But the enteric nervous system is really, really important. You couldn't live without it. You can live without your brain, and people do. But without your gut, you would die fairly quickly. It's got 500 million neurons, so it's more neurons than your spinal cord. It's 30 feet long. You know, it would stretch up to a 3-story building. It's got 2 layers of skin and muscle and nerve in there. It can operate entirely independently of the central nervous system. So even if you were to separate it from the central nervous system completely, it would still mostly just chug along quite happily, digesting whatever you put into it. It has the same reflexes as the central nervous system. It uses the same neurotransmitters as the central nervous system. It's affected by the same drugs as the central nervous system. So I like to joke that nicotine and coffee get all the nervous systems moving. It contains 90% of the serotonin and 50% of the dopamine in the body. These are obviously neurotransmitters, which are very important and highly talked about in the central nervous system. And it's often called the second brain. This is a great book by medical doctor Michael Gershon. And he calls the enteric nervous system the second brain just to highlight its importance. But I hope that by the end of this talk, I'll have convinced some of you that it may be the first brain. So if you're looking at the evolution of nervous systems, perhaps a question you want to ask early on is, why do we have them in the first place? As far back as Aristotle, philosophers understood that movement is the most essential part of life. Charles Sherrington, who's a personal scientific hero of mine, he studied movement and reflexes. He coined a ton of terms we use. He coined the term synapse, for example. And this, I found this quote so profound. To move things is all mankind can do. For such, the sole executant is muscle, whether in whispering a syllable or felling a forest. Movement is the only way we can interact with the world. This is a picture you don't often see of Charles Sherrington. This is him when he's about my age at his postdoc in Cambridge, and he's outside his lab with his colleagues smoking a cigarette. Aldous Huxley, of the famous Huxley family, Hodgkin and Huxley was related. Thomas Huxley, Darwin's friend, was related. Consciousness is only possible through change, change is only possible through movement. And in the modern world, Daniel Wolpert, great neuroscientist, says that we have a brain for one reason and one reason only, and that's to produce adaptable and complex movements. And I can't say this any better than he can, so I'm going to turn the floor over to him for about 2 minutes to talk about this.
I'm a neuroscientist, and in neuroscience, we have to deal with many difficult questions about the brain. But I want to start with the easiest question, and the question you really should have all asked yourself at some point in your life, because it's a fundamental question if we want to understand brain function. And that is, why do we and other animals have brains? Not all species on our planet have brains, so if we want to know what the brain is for, let's think about why we evolved one. Now, you may reason that we have one to perceive the world or to think, and that's completely wrong. If you think about this question for any length of time, it's blindingly obvious why we have a brain. We have a brain for one reason and one reason only, and that's to produce adaptable and complex movements. There is no other reason to have a brain. Think about it. Movement is the only way you have of affecting the world around you. That's not quite true. There's one other way, and that's through sweating. But apart from that, everything else goes through contractions of muscles. So think about communication, speech, gestures, writing, sign language. They're all mediated through contractions of your muscles. So it's really important to remember that sensory, memory, and cognitive processes are all important, but they're only important to either drive or suppress future movements. There can be no evolutionary advantage to laying down memories of childhood or perceiving the color of a rose if it doesn't affect the way you're going to move later in life. Now, for those who don't believe this argument, we have trees and grass on our planet without the brain, but the clinching evidence is this animal here, the humble sea squirt. Rudimentary animal, has a nervous system, swims around in the ocean in its juvenile life, and on some point in its life, it implants on a rock, and the first thing it does in implanting on that rock, which it never leaves, is to digest its own brain and nervous system for food. So once you don't need to move, you don't need the luxury of that brain. And this animal is often taken as an analogy to what happens in universities when professors get tenure, but that's a different issue.
So yeah, he explains it very well, and I didn't want to plagiarize him, so I just threw in a couple minutes of him talking about that. But he doesn't have quite the nuanced explanation that's necessary for this. For example, he does not go any further than why do we have nervous systems? Well, we have them to move. To me, the obvious next question is, well, why? Why do we move? Not all multicellular species move on the same fast timescales that we animals do. Plants and fungi obviously move, but they move very slowly. This is a time-lapse video of a plant growing, and it's moving through the world to put itself in a nice place. This is a sponge, which doesn't move in the world, but it does move the world through itself. So yeah, giving this a lot of thought. I went back to my days in the Boy Scouts, and we learned about the survival rule of threes, which is we can live for about 3 minutes without air, about 3 hours without shelter in a harsh environment, about 3 days without water, about 3 weeks without food. And so we animals have to move quickly because we have to meet our survival needs by moving throughout the world. And this just came to me recently. Can anybody think of any plant species that move quickly? Perhaps more quickly than animals? Carnivorous plants do. So Venus flytraps move more quickly than flies. Why do they do that? In order to eat. They live in very protein poor environments. So in order to get the protein they need to live, they have to eat flies, and they have to move very quickly to do that. And these carnivorous plants actually use calcium action potentials to activate their rapid movements, much like animals have developed nervous systems. So that's a nice, to me, it's a nice exception that proves the rule. So let's talk about early animal evolution. So obviously you need air, water, and food in order to survive. Animals evolved in the oceans where air and water were free. As long as you're small enough, oxygen will just diffuse through you. You don't even have to worry about it. But so then the limiting factor then becomes food. Animals have to move to eat, and they have to eat to move. So let's go back to Daniel Wolpert's example of the sea squirts quickly and add a little bit of nuance. So yeah, as he said they were rudimentary animals, and that's not really true. They're actually chordates, so they're fairly closely related to us. When they're young, they have a notochord and a nerve cord and a sort of a little brain at the top and a gut and all this stuff. And they swim throughout the world to find a nice place to live, and then they settle down. And then it's true, they do digest most of their central nervous system. They no longer need to move through the world, and so there's no need for it. But they do still have to move the world through themselves, and they do still have to react to predators coming at them, and they do have to, so they kept some nervous systems. They've got a nervous system in their gut, which they kept, and they kept their peripheral nervous system, which interacts with a much reduced brain in order to respond to environmental threats and things. Yeah. So yeah, let's just summarize. I'm going to do this all the time to talk. Vertebrates, we have multiple nervous systems. We have them because we have to move. We have to move because we have to eat. The next question you might want to ask yourself is, well, how do animals move? And the oldest and still, I would say, commonest form of multicellular movement in animals is called peristalsis. And this is waves of muscular activity in muscular tubes. Okay, so animals, almost all of them, I believe all of them, have two layers of muscle in their body. A layer of circular muscle, which wraps around the length of them, and a layer of longitudinal muscle, which stretches along the length of them. And with waves of activity traveling through those two layers of muscle, you can get peristalsis, which can move can move a worm through the earth, or it can also move the earth through a worm. So inside us, in our guts, we still use peristalsis to this day. These two layers of muscle and waves of activity traveling through them to move stuff through us. Yeah, so if you contract all the circular muscles, your tube is going to get long and skinny. All the longitudinal muscles is going to get short and fat, and you can use it to move.
Cool though. Very cool though.
And okay, so it turns out that animals are mostly made up of tubes. And so our digestive system, obviously a long tube. Our cardiovascular system, our heart, is actually two tubes that wrapped around each other and became heavily muscularized. Our respiratory system, our lungs, are just a set of branching tubes. Reproductive system, lymph system, auditory system, the integumentary system, which is the skin around our bodies, these are all tubes. Even 3 of our 4 vertebrate nervous systems are very clearly tubes. The enteric nervous system is the nervous system in the tube of the gut. The peripheral nervous system is the nervous system in the tube of the body. And the central nervous system is a hollow tube with ventricles at the top, which connect to a central canal. And you've got cerebrospinal fluid constantly flowing through this tube. So again, we have multiple nervous systems because we have to move, because we have to eat. We move using waves of activity in muscular tubes, and we're mostly made up of tubes. Even 3 of our 4 nervous systems are very clearly tubes. So you might start to wonder, oh, where do all these different tubes come from? Do they share common features, or is each one unique? Was there one original tube, or did they all evolve independently? So in terms of where all the tubes come from, if there's no other take-home message you get today, know that the gut is an amazingly adaptable tube. And this stuff is all known already. This is nothing new from me. Very, very little in this is new from me. This is just a reinterpretation of existing facts. But yeah, so the esophagus, stomach, intestines obviously are just specializations of the gut tube. It turns out that our lungs are a branch off of the gut tube which then goes on to branch and branch and branch and become specialized to process oxygen. Our liver, pancreas, gallbladder, and appendix are just diverticula of the gut, which then become highly specialized to do other things. Even our urogenital system is just comes from the gut. Our notochord, the stiffening rod in our backs, early animals' backs, still exists in us to this day, bits of it, as the intervertebral discs. Those are actually the original notochord now that live in between the vertebrae. That is just a blurb off of the gut, which then becomes this stiffening rod down the length of chordates. Our auditory tubes and our middle ear are literally just branch off the gut to become our hearing apparatus. One of the coolest things is that the three middle ear bones in our ears are actually ancient fish jaw bones that have been repurposed to become our hearing apparatus to transduce sound between air and fluid. So yeah, the gut is an amazingly adaptable tube. In terms of all these different tubes sharing common features, yes, I've looked at all of them. And I coined the term swim. And a little nerd joke here, the C is silent like in Nidarian. And that'll come up later, I hope. But yeah, so these swim tubes, they all share these features. They are cuticle producing. So your body tube on the outside can produce nails, claws, hair, scales, feathers, armor. I'm calling all these things cuticle. There might be a better term. Cuticle producing skin covered. The tubes are covered with an epithelial layer, which has a basal membrane at the base of it, which is waterproofable. Not all of them are waterproof. Your capillaries, you want them to be leaky, but they are waterproofable. They are innervated and they are musculoconnective. And these tubes have two layers of musculoconnective tissue, as we talked about in peristalsis, a circular layer and a longitudinal layer. And I say musculoconnective instead of muscular because some of them just became connective. So in the lungs, for example, you've got sort of circular, smooth muscle surrounded by longitudinal cartilage. In the heart and arteries, you've got circular, the tunica media, circular, surrounded by the outside, which is more longitudinal. The gut has a circular layer of muscle inside a longitudinal layer of muscle. The body, this is the body of a worm, but it has longitudinal muscle inside a layer of circular muscle, and that is inverted compared to the rest of the tubes. The body tube is somehow radially inverted compared to the rest of the tubes. So yeah, I'm going to introduce my adaptable gut hypothesis at this point. I'm going to say the tube is the basic building block of animals. The earliest animals were a single tube. Jellyfish, hydra, coral. The Nidarians are a single tube with two layers. Our gut is the archetypal tube, and I think our gut is the same as those earliest animals. I believe our body tube and the body tube of tube inside a tube animals is the gut tube wrapped around itself and connected at the other end. Most of the rest of the body is more tubes. The organs, once you have a tube inside a tube, you've got an intraperitoneal cavity where you can grow lungs and you can grow a heart and you can do different interesting things. As a single tube, you can't grow lungs. You'd have to drag them around in the world. You need this internal place where you can develop new organs. So yeah, so this is a jellyfish. This is a hydra. These are Nidarians. These are just a single tube with two layers. Later in evolution, this is an echinoderm, which is fairly closely related to us. And its outside body happens to be translucent. So you can see the outside body is a tube, which is wrapped around that gut tube. And I think the way it got that way is the gut tube wrapped around itself and connected to become the body tube with space in between those two tubes. So in terms of the evolutionary origin of brains, I believe the enteric nervous system is the first nervous system. It's the same nervous system that the hydra and jellyfish have. I believe the peripheral nervous system or the somatic nervous system is the second nervous system, which is a direct copy of the enteric nervous system because the body is a copy of the gut wrapped around itself. The peripheral nervous system originally was an exact copy of the enteric nervous system. The central nervous system is the body tube inverted into itself. And this is already well known. You can see this and I'll show pictures in a little while, but that would make it a second generation copy of the enteric nervous system. It would make it an inverted copy of the enteric nervous system, which means it's a non-inverted copy of the enteric nervous system, which explains a lot of, there's a lot of amazing similarities between the central nervous system and the enteric nervous system, and I'll get to The autonomic nervous system, in particular the sympathetic nervous system, I believe, is a highly modified tube, which is also the body tube inverted into itself. And this is a brand new idea. And it's very young and speculative, but I hope to get feedback and everything from people. So yeah, this is the essence of the adaptable gut hypothesis. The original animals are a single tube with 2 layers, a jellyfish, hydra, coral. To become the tube inside a tube anatomy of animals, that gut tube wrapped around itself and connected at the other end. And this is magical, because hydra, jellyfish, corals obviously can't develop armor, because their body is their gut is their body, and they have to be able to move to digest. But once you've got a digestive tube inside a body tube, this can stay soft and squishy and digestive. And the outer body can now do cool things. It can develop stiffness and armor and protection. I believe this is the most plausible explanation for the Cambrian explosion that I've ever heard. In the Precambrian, very few animals fossilized, because there's no armor, there's no stiffness, there's no nothing. These were soft gut tubes. At the Cambrian, all of a sudden fossils are everywhere, and every single one of them is a tube inside a tube anatomy. Well, anyone with armor and stiffness. The most common ones are the ones that have this tube inside a tube anatomy, because the outer body can now get stiffness and armor and protection because it's got the soft, squishy, digestive gut in the middle of it. To get to the central nervous system, the brain and spinal cord, there's a primitive, not primitive, but an old animal called Amphioxus. It's kind of fish-like, and until it's adult, it has a hole right in its head. As an adult, that hole eventually closes. But I guess essentially it was probably like a second sensory apparatus. You know, you can get 2 layers of muscle. You can pull water in and out. It's kind of like smelling the seawater or whatever. Evolutionarily, it eventually goes on to dump into the gut. It's called the neuroenteric canal. And in humans, even to this day, you can still see this process in human embryology. Our brains start as a hole in our head that dumps in our gut. If the bottom end doesn't close, you end up with spina bifida. If the top end doesn't close, you end up with anencephaly. Your brain never develops. And these are obviously very traumatic embryological problems. But for most vertebrates, they end up closing off successfully. And now you've got an entirely separate central nervous system in the body. The autonomic nervous system, the sympathetic nervous system, sits on the back wall of your trunk, sort of in front of your spinal cord, but behind all your organs. And it sits exactly where this Amphioxus animal developed an atrium. It pulled its body tube into its own body and wrapped around things we'll get to the details in a little bit. But it happens to sit exactly where you find the the sympathetic nervous system. And it also happens to sit exactly where we find our internal skeletons, okay? So exoskeletons are easy. Crabs, mollusks, insects, anybody can do an exoskeleton, but very few species have the ability or develop endoskeletons like us. And I think this might be a good explanation for it. And I'll get into more details a little bit. I do want to talk a little bit more about how the gut and the body tubes are radially inverted, because to me, this is one of the strongest pieces of evidence for this idea. If we look at our gut wrapping around itself to become a gut and a body, we can see that the inside of the gut tube becomes the outside of the body tube. And the outside of the gut tube becomes the inside of the body tube. They are radially inverted, okay? This adaptable gut hypothesis immediately explains this. And so, yeah, so in the innermost part of our gut, we've got a gastrodermis, then circular muscle, then longitudinal muscle, then a peritoneum, a smooth epithelium that can allow organs to slide around. In the body, on the outside is where we have the dermis. We have an epidermis on the outside of our body. Then we have circular muscle, then we have longitudinal muscle, and a peritoneum innermost in the body. And yeah, so even in humans to this day, we can see that the deepest muscles of our body are those longitudinal ones. And the most superficial muscles of our body are circular in that they're more perpendicular to the length of the body axis. It's very easy to see this in worms because it's very clear that the circular muscle layer is outside the longitudinal of the body, and the circular layer is inside the longitudinal layer in the gut. Yes, so I'm not a geneticist, and I don't know very much about genetics at all. But I knew that this would be an immediate way to rule this idea out if it was wrong, because if the enteric nervous system and the central nervous system use completely different genetic mechanisms to develop themselves, then yeah, this idea is immediately and absolutely wrong. Adaptable gut requires that the enteric nervous system was the first to develop, and then the peripheral nervous system, and then the central nervous system. Which means that you would expect that they would all be based on the enteric nervous system development genetics. And it turns out they are very similar. So genetics do not rule out this idea. To arrange these nervous systems in the anterior posterior direction, or rostrocaudal direction, Hox genes are used, this family of genes called Hox genes. And that, you have to have your esophagus before your stomach, before your liver, before your intestines. You have to have your brain before your midbrain, before your hindbrain, before your spinal cord. And these Hox genes are what lay out the nervous systems in this direction. In the other direction, the direction perpendicular to the body axis, both the enteric nervous system and the central nervous system use the same gene products to develop themselves. So they use sonic hedgehog and bone morphogenetic protein 4. They use gradients, opposing gradients of these gene products to develop themselves in this direction. So you want to have your circular inside your longitudinal in the gut. You want to have the dermis on the inside, not the outside of the gut. In the central nervous system, it doesn't have to be radially symmetric, and it's not, but they still use opposing gradients of sonic hedgehog coming from the notochord and BMP4 in the outside body to arrange, you want your motor neurons first, and then your V2, your sensory neurons, as you move towards the back of your body. So yeah, so in both the longitudinal and opposite directions, both of these nervous systems use the same genetic programs to do it. Although they're slightly different, as you would expect over time, but it seems clear that they probably evolved from the same place. So I believe that adaptable gut has a lot of explanatory power, far more than our existing understanding of how these things happen. It immediately explains the tube inside a tube anatomy of us later evolved animals. It immediately explains the coelom. Coelom is an old term for the intraperitoneal cavity, the space between those two tubes. This immediately explains how we got that. It explains the inversion between the gut and the body tubes. It's a plausible explanation for the Cambrian explosion. It simplifies our evolutionary understanding of muscle, and I'll get to this in a little bit, but this is kind of fun. Yeah, and it could explain why we have an endoskeleton as vertebrates instead of exoskeletons. It would be an internalized exoskeleton, and we wouldn't be the first to do this. Squid and cuttlefish both have internal skeletons, which are actually internalized exoskeletons. They're shells. They internalize into their own body to get an endoskeleton. And I'm saying we vertebrates, I think we did the same thing. Neural crest and the autonomic nervous system as an invagination of the body tube, and I'll get into more details on this briefly. It immediately explains the enormous similarities between the enteric nervous system and the central nervous system. And at the very end of the talk, I'll talk about some implications for neuroscience. I believe it implies that pattern generation is waves of activity traveling through nervous systems, much like peristalsis is. So yeah, an idea like this, you really gotta dot your I's and cross your T's and look into everything. So I went back into deep history to see maybe I could figure out where it went wrong, or maybe I can see where somebody already had this idea and I'm wrong. So yeah, I went way back in time, and I'm gonna just run you quickly through the evolution and development of evo-devo, which is a field in biology which studies the interesting correlations between evolution and embryological development of animals. So way back in 1817, Christian Pander, a wealthy German scientist, young, was examining, he could buy the latest technology. He had the latest microscopes, which by today's standards are horribly primitive. But at the time, it was like me having an electron microscope in my apartment. And he studied chicken embryos, basically, eggs like we eat for breakfast. And yeah, so the years that I think things went wrong are bolded and underlined here. And I think one of the problems is went wrong right from the very beginning is because Pander saw layers instead of tubes. And I think I know why. And I'm sorry for the resolution, but this is the best quality of this image I could find. This is, the top here is a chicken embryo, okay? And it sits on top of this absolutely enormous yolk, which is evolutionarily very new. You know, it's very different from animals that came before it. And yeah, so he would cut the embryo off of that enormous yolk and macerate that and cut it in different ways and look at it under the microscope. And when he did that, you know, he saw it as three layers. He called this inner layer the endoderm, or inside skin. He called the outer layer the ectoderm, or the outside skin. And he called everything else in between mesoderm. Okay. But if you take out that evolutionarily very new, enormous yolk from a chicken embryo and you pull the edges together, then it looks just like a frog embryo, which is very clearly tubes. Okay. So from the very beginning of our understanding of this, we kind of went awry because we saw it as layers instead of tubes. Oh yeah, one thing I did want to point out here, just in terms of time scale, is that Darwin didn't publish The Origin of Species until 1859. We didn't even have a cell theory until 1839, right? So he's working, he doesn't have much to work with. So there, I'll talk about him a little bit. Rathke took this germ layer theory and extended it to arthropods. He said, yeah, when I look at arthropod embryos, I see the same layers. Von Baer extended it to all vertebrates. He looked at a variety of vertebrates and he said, yeah, I can see this layer theory working. But he saw 4 layers, not 3. Then in 1849, Huxley extends germ layer theory to cnidarians, which are the jellyfish, the hydra, the corals, only 2-layered animals. So in 1849, Huxley says, well, I see layers in my evolutionarily older animals that I'm exploring. These guys have 2 layers. These guys have 4 layers like us. You know, there's 2 layers in the gut and 2 layers in the body. But originally they just had 2 layers. And he extended it to cnidaria. And he said, so basically the inside skin of a hydra is the same as the inside skin of our gut. The outside skin of a hydra is the same as the outside skin of our body. And then mesoderm, this magical 3rd layer, fills in everything between our gut and our body. I think thinking of these things as tubes is a much simpler, more concise, more parsimonious explanation. But yeah, so we end up just having 3 germ layers: endoderm, the inside skin; ectoderm, the outside skin; and mesoderm, which is magical and fills everything else in. Darwin publishes The Origin of Species. Haeckel, who's got a bad name these days, and he shouldn't because he's awesome, develops recapitulation and gastria theory. Gastrulation is very important, and we'll talk about that in just a little while. He also sees 4 layers. And then in 1873, Lancaster, a very famous British biologist, physiologist, he actually names the classification of animals according to how many germ layers they have. So up to this day, we still refer to cnidarians as diploblasts, 2 germ layers. And us and everything like us with muscle and stuff is triploblasts, or 3 germ layers. But this is bad because he's baking a theory into the classification of animals. He's begging the question. It's circular logic. And we've been stuck with this crazy idea that we can only have 3 germ layers ever since. And the germ layers they're not even internally consistent, right? So according to germ layer theory, the endoderm, or the skin lining the gut, is just the skin and the basal membrane of the gut. The muscle layers of the gut and the peritoneum of the gut, that comes from mesoderm. They say the ectoderm is just the outside skin of the body and its basal membrane, not the muscles underneath, not the peritoneum underneath, just the outside skin. But somehow ectoderm becomes the entire central nervous system. It becomes, with its multiple layers, all its meninges, with all that stuff. Somehow this ectoderm is very different from the endoderm because it can become the central nervous system. And then the magical mesoderm fills everything else in. But you can see that as soon as they have mesoderm, they immediately have to split it into somatic mesoderm and splanchnic mesoderm. That is the mesoderm of the body and the mesoderm of the gut. And I'm saying mesoderm is a chimera. It's a phantom. It doesn't exist. This somatic mesoderm was always part of the outside body tube. This splanchnic mesoderm was always part of the inside body tube. Yeah. So if you press someone who knows a lot about germ layer theory, they'll say, well, the endoderm isn't just the skin lining the gut. It's also the skin lining the gut-associated organs. But that's all it is. The ectoderm is, the skin lining the outside body, and also remarkably somehow the entire central nervous system. And mesoderm is magic, and it fills in muscle and blood and bone and everything else. It's not, to me, it's not a satisfying explanation. Oh yeah, mesoderm. So that was 1873. Lancaster locks us in, bakes us in to germ layer theory thinking by naming animals after germ layer theory. And I want to point out that it's not even like there's a unified theory of animal evolution. You have to have germ layer theory to kind of explain the bulk of things. Then you've got concrescence theory, which was used to help explain why the central nervous system becomes part of the body tube. The Hertwig brothers in Germany developed coelom theory to explain why we have the intraperitoneal cavity. You have to pull in lots of different theories to explain animal evolution. There's no unified theory of animal evolution. In 1881, Kleinenberg, a Russian scientist, felt like I did. He denied the existence of a separate, unique mesoderm. And I haven't been able to read him in his papers, just summaries. They're in Russian, I believe, or German. It denies the existence of a separate mesoderm. He believes like I believed. You gotta, earliest animals are 2 layers, but the later layers, they're not magical mesoderm. That doesn't explain anything. In 1908, Gaskell, a famous physiologist, American, developed an early adaptable gut theory. It's kind of fun. I'll talk about that in a little while. In 1999, Brian K. Hall, Canadian, has made a very strong case that if we're going to have germ layers, neural crest should be considered a 4th germ layer. The neural crest is when the central nervous system tube closes. Around that time, a blurb of cells come off and migrate all throughout the body to do interesting things. They make your face, they make your skeleton, they make your adrenal glands, they make your sympathetic nervous system. It does all these incredible things. And so, if we're going to have germ layers, it should be another one. He doesn't seem to have gotten much traction on that. And in 2017, exactly 200 years after the original publication of germ layer theory, I presented a poster at Society for Neuroscience. I really wanted to hit that year. I wanted to publish that year, but this stuff takes a long time. I'm going to talk about coelom theory quickly because in biology textbooks, you're going to have two holy trinities. You got the holy trinity of germ layer theory. An animal's either a homoblast, like a sponge, no germ layers, a diploblast, like a cnidarian, jellyfish hydrocoral with 2 layers, or a triploblast with 3 layers, like us and everything evolutionarily newer than the cnidarians. And you'll also see coelom theory with a holy trinity of acoelomate. The earliest animals do not have an intraperitoneal space. They are acoelomate. They have no coelom. Roundworms, they call pseudo-coelomate. They do kind of have a coelomic cavity, kind of an intraperitoneal space, but it's not lined by skin and it doesn't have the layers of muscle. So they call it a pseudo-coelom, a fake intraperitoneal cavity. And then you've got the eucoelomates, or just the coelomates like us, which have outer body tube with all the layers in the peritoneum, an inner body tube with all the layers and lined by a peritoneum, and then this peritoneal cavity or coelom where we can grow new organs. So I'm going to quickly run you through the phylogeny of this idea. If I can't sort of trace this through our living relatives, the idea is not going to work. So I'm going to run you through the evolution of animals in just a few minutes here. Quadflagellates are sort of the origin of multicellularity. Sponges and the cnidarians with 2 layers. These guys are okay. Well, yeah, we'll get there. But this is just to show you. And there's different ways to interpret. There's no. It's very difficult to build family trees, and so there's different ways to look at it. But I'll walk you through this. Just to get to the point where we became multicellular, life had already been evolving for billions of years. So yeah, the Big Bang, 14 billion years ago, Earth 4.6 billion years ago, origin of life around here. And then we have to get all the way up here just to get to eukaryotes. And Lynn Margulis came up with the idea of endosymbiosis, which is that mitochondria and our other organelles were actually invaginated bacteria, which did useful things for us. And we developed this cooperation, collaboration, symbiosis with these bacteria, which made us much more successful, them and us, much more successful in life. At this point, almost all of life is single-celled, but there are many experiments taking place. And one of those evolutionary experiments led to us. So yeah, multicellular animals is only about 600 million years. So yeah, choanoflagellates. This is the link between single-celled life, single-celled animals, and us multicellular animals. They're called choanoflagellates. They have a flagellum, which they can swing around either to move through the world, or if they're sitting still, to move water around them. And then they've got a choano, a sticky collar, which catches bacteria, which are then endocytosed into the cell body to eat them. So yeah, so one end is a digestive end, and one end is like an extracellular matrix making end. And yeah, the only reason that we can be the amazing multicellular animals that we are is because each one of our cells is an amazing animal on its own. So these guys already have like a primitive sodium-potassium pump. They have calcium and sodium channels. They have these genes for adhesion to stick to other cells, for producing extracellular matrix, for signaling, for talking to other cells. These are already existent in each and every cell of us. We are like an enormous extended family of each one of us individually. It's like an enormous extended family of choanoflagellates who decided to live together and division of labor and all that. And yeah, so to explain the morphological changes during evolution, I came up with what I call the water balloon model of early animal evolution. So when times are good, these guys don't have to split up when they reproduce. They can hang out as a family. And you'll see that many of them, some of them, will turn into these spheres, these colonial spheres, with the digestive ends all pointing outwards and the connective ends all pointing inwards, sort of to the inside of the sphere. So you can picture this as a water balloon, single layer with all the digestive stuff outwards and the connective stuff inwards. Sponges came next evolutionarily on the way to us. And yeah, so this is where gastrulation starts, the origin of the digestive tube. So if you've got that water balloon, if you can imagine shoving your finger down inside it to create a tube, now you've got the first tube. And this is really good for digestion because it's very expensive to produce acid and the digestive juices and all that stuff. And if it's all on the outside of a sphere in the ocean, water can wash it away. If it's on the inside of a tube, you can produce all the acid you want. It'll stay right there. So this, yeah, obviously helped. You start to see division of labor, specialization of cell types. And these sponges do not have muscle fibers, but they are capable of slow movement. There is some actin myosin sort of around that allows them to very, very, very slow move. And yeah, so this is the inside of a sponge. This is not choanoflagellates. These are sponge cells. But obviously, it seems quite clear to everyone that they evolved from the choanoflagellates. Moving up, the cnidarians, the jellyfish, the hydra, the corals. So this is, again, water balloon wise, a hydra is just putting a finger down into a water balloon, and then the water balloon itself sort of extending tentacles out, each with 2 layers of it's just 2 layers, 2 epithelial layers. But this is, to me, the simplest explanation of how muscle evolved. In layer theory, these guys don't have muscle. They don't have anything like muscle. Muscle can only come from mesoderm, and these guys don't have mesoderm. So yeah, there's no muscle here, nothing to see here. But these guys have They don't have a true epithelium. They've got, it's called either myoepithelial cells or epitheliomuscular cells. They've got an epithelium up here, but at the base of it, they've got these longitudinal muscle fibrils on the outside of their body. And on the inside of the body, at the base of the epithelial cells, they've got these circular muscle fibrils, just like in our gut, circular inside, longitudinal. But they're not pure muscle. They're not pure epithelium. Yeah, to me, this is how muscle evolved. And obviously, muscle and nerve had to co-evolve. Muscle is useless without a way to activate it. And neurons very clearly are just epithelial cells that became highly specialized. And then the water balloon model wise, again, the hydra is putting a finger into it. The jellyfish are like taking the water balloon and squishing it up and making it concave. And then you can pull down tentacles out of there, and all the digestive stuff is on that side, the protective stuff's on the outside. So moving later in evolution, worms appear. And this is a worm where the two holy trinities break down, because these guys clearly have muscle, so clearly have mesoderm, but they are acoelomate. So they're like the second level of germ layer theory, but still the bottom level of coelom theory. Two muscle layers, circular inside, longitudinal outside. Lots of specialization of cell types. And one of the craziest things these animals do is this is actually the entire length of its body. You know, its body is this long. What it does is it'll pull 2/3 of its body into its own body. It's called a proboscis. And they use this as a way to eat, as a way to catch prey. So it's like spring loaded, boom, stick it out with a little hook at the end, maybe some neurotoxin, get your prey. And then you can pull that around to where your mouth is, okay? And so water balloon model wise, this is like taking that water balloon and pushing your finger all the way through and then popping it all the way through the other side. So now you've got a through gut. And through guts are hugely advantageous, right? Because early digestion is not the same as later digestion. It's great if you can specialize digestion along the length of a through gut. This is where through guts appeared. And now this is the adaptable gut hypothesis. Now you've got, you got the single water balloon with a hole all the way through it. Now that water balloon has to wrap around itself again and attach at the other end to get an endotube and an ectotube, if you will, a gut tube and a body tube. So these are annelids, like earthworms, and they have 2 layers of muscle, 2 layers of skin in the gut, circular inside, longitudinal in the gut, 2 layers of muscle, 2 layers of skin in the body, circular outside, longitudinal in the body. So again, they're radially inverted. Segmentation appears at this point, which is hugely important for us more complex animals. Segmentation allows, repeatable units that can then specialize. So forebrain, midbrain, hindbrain, it all comes from this original segmentation. A very cool thing happened at this point, which turned out to be good for us. These animals have a pharynx, which can come out of its body. So again, this is its whole body tube. But what some of these worms can do is they can pull their pharynx into the rest of their body and then pop it out like Alien to eat prey and stuff. So they've got this pharynx, which is outer body tube, pulled into their bodies. And that's, in my mind, and I think other people as well, this is where we got our pharynxes and our voice boxes. And this is why our outside brain controls what is an inside part of us. So good. Priapulida, penis worms. Priapus is the Greco-Roman god of erections. These guys are both tripeloblastic. They clearly have muscles, so they've got 3 germ layers, and they're coelomate. So they're a tube inside a tube, like the annelids we just talked about. Again, gut and body tubes radially inverted. And this animal this species will invert its own body into its own body for defensive purposes. So it's already a tube inside a tube. It pulls that tube inside a tube inside the tube inside a tube, and so on. So this sort of crazy tubular movement. The point I'm trying to make is that this crazy tubular stuff is not at all abnormal. Okay, so later in evolution, you've had the gut tube inside the body tube for some time now. And the body tube has had a lot of time to specialize. Armor, limbs, all these different things, sensory apparatus, antennae, eyes. So yeah, these are, they've got true muscle, they're triploblastic. And okay, yeah. So they have, an important point here is that there are ring brains, okay? The gut goes right through the middle of their brains twice. So this is their brain. You can see the gut has to go right through the middle of it. And again, at the anus, the gut has to go right through the middle of the nervous tissue. And yeah, you can see that. You can see why that is, because if you got a tube inside a tube, the only physical place where those two tubes meet is in rings at either end. So this is where you get ring brains. This is where the coalescence of nervous tissue in these rings ultimately led to our brains. Yeah, these guys and most animals before chordates put their centralized nervous system on their belly, which makes a lot of sense, because if a predator breaks your nervous system, all your muscles are useless. So you'd want it in the most defended spot. We have it on our back, and we'll get to that. Okay, so yeah, we've gone through sponges, cnidarians, the Nemertians, some of this. Now there's a huge break, a leap in evolution between protostomes, what are called protostomes, and what are called deuterostomes. And in terms of genetics, in terms of evolution, in terms of all this evidence, these are obviously very separate groups. And so you have to come up with a way to name these groups. And yeah, I'm not impressed with the names that have been chosen. Protostome means first mouth. Okay, so the earlier animals to evolve are all protostomes. When you gastrulate, you turn from that water balloon and you push a gut inside it. That's called gastrulation. That hole becomes their mouth. Or if they don't have a through gut, it's both their mouth and their anus. But yeah, their mouth. They say that deuterostome means second mouth. And so in general, it's true. The first invagination into the water balloon becomes the anus instead of the mouth. And then the mouth appears later. But this is not true across deuterostomes. There are many, many exceptions. And to me, it's not even the most interesting thing about deuterostomes. The most interesting thing about deuterostomes is these hydropores. Okay, these are invaginations of the body tube into the body with 2 layers of muscle, 2 layers of skin. And so, yeah, so this is, Haeckel drew this like a generic deuterostome. But yeah, a perfect example of this is the starfish. They use tube feet to move around in the world. And these tube feet are nothing but massive numbers of invaginations of the body tube into itself. So each one of these guys has 2 layers of muscle, 2 layers of skin, and can move water in and out. And this is how starfish get around in the world. Hemichordates are kind of half chordates. I just bring this up because it's a nice transition between the earlier animals, which have their central nervous system on their belly, and chordates. We have our central nervous systems on our backs. And it's quite obvious. Geoffroy Saint-Hilaire in 1822 came up with the fact that, wow, we must be somehow inverted copies of insects. Yeah, this inversion also in the timeline of evolution seems to me to occur at the exact same time as when our brains became cross-wired. So our left side of our brain controls the right side of our body and vice versa. In worms, that's not the case. Why would you do that? But we have our brains cross-wired. And this happens to be around the same time that the bodies had to invert it anyways. So there's a lot of this is ripe for research, for further exploration. Talk about a little more about ring brains. All animals older than cephalochordates have their guts right through the middle of their brains. But you can't become vertebrates if that's the case, because you would have to have a donut-shaped skull with your gut going right through the middle of it. It just wouldn't work. And so somehow the vertebrates had to break their gut out of their head brains. And the vagus and the sacral part, I think I'll get to that a little more later. And I'm going to try to wrap this up quickly. I'm sorry. So in cephalochordates, a lot of things appear very quickly. They get a new mouth. So you don't want your gut going right through the middle of your brain. How do you get around that? grow a new mouth. Amphioxus and in us, apparently, our new mouth is an ancient gill slit that got repurposed to be a mouth. That allowed it to bypass the central nervous system. I believe the ring brain that originally, that our guts originally went through is now the vagus nerve, but that's highly speculative. So yeah, they get a new mouth, they get a notochord. So if you're a worm, it's very hard to swim because you don't have anything to pull your muscles against. A stiff rod down the middle of your body would be very useful. And this is when the notochord appears, which is just a blurb off of the gut that stiffens, becomes this stiffening rod that allows much better movement through water. And this is where the atrium appears. So these amphioxus, they live in sand tubes, and they're filter feeders. They don't want to take in water and have them go right out their gills and stir up sand everywhere. They want to be able to control their water intake and output. So as deuterostomes, they're able to invaginate their body tube into their tube. And they did this, they form an atrium. The body tube invaginates in all the way around their gills and all their organs in order to form this atrium where the water comes in, goes out the gills, now it's in this atrium, and they can push it out whenever they want through this atrial pore. And yes. So yeah, now we're moving up to vertebrates from cephalochordates. A lot of things appear very quickly. Endoskeletons, the neural crust, the endocrine system, the adrenal glands, the autonomic nervous system, our faces. And yeah, so this is, again, highly speculative. Perhaps the most speculative part of my presentation is that this atrium in Amphioxus is what became all those things. And it's an invagination of the body tube, 2 layers of skin. Glands are epithelial in origin. Perhaps the skin from this tube became our adrenal glands. Perhaps the nervous tissue from this invagination became our sympathetic nervous system. It would sit exactly up against the back wall of the body where the sympathetic nervous system is. And if it wrapped all the way around, you would have vertebrae and ribs. And again, I said exoskeletons are easy. Anybody can do an exoskeleton. So, it's easy to make armor or stiffness on this outside. So you can imagine if this were invaginated into the body and sitting next to each other, now you've got 2 layers of exoskeleton producing material, exactly where we find our endoskeleton. So to me, this is very, very intriguing. And that is adaptable gut hypothesis. The enteric nervous system was first, wrapped around itself to become the enteric and the peripheral nervous system. The central nervous system is the body tube invaginated into itself. Later, we get the autonomic nervous system, the sympathetic nervous system, which I believe is another invagination of the body tube. And just really quickly, sorry to delay, Gaskell, who was actually Charles Sherrington, considered him his primary mentor in life. And I only figured this out when I started researching this stuff. He wrote, he had this early adaptable gut hypothesis. And he wrote an entire book about it called The Origin of Vertebrates. He saw the remarkable similarities between the central nervous system and the enteric nervous system. Okay, this is a drawing from his book. And just like I've seen, wow, they're really, really similar. What's going on here? Yeah, so he proposed actually that the enteric nervous system directly became the central nervous system. So he thought, oh, well, animals just transformed their gut into their central nervous system, and then they grew a new gut. But as we've talked about, animals always have to be eating. You can't lose a gut to get a central nervous system. You always have to have a gut. So yeah, this idea died on the vine. But he did write a letter to Huxley. This has helped me, because this is crazy, and I don't know if it's going to go anywhere. Huxley wrote back a very nice letter to him, and he said, "Go on and prosper. There's nothing so useful in science as one of those earthquake hypotheses which oblige one to face the possibility that the solidest looking structures may collapse." And yeah, this should be the final slide. Potential implications, movement is the reason, it's understudied. Neural patterns as traveling waves of activity. This is really cool, so I'm just gonna spend a little bit of time on this. Peristalsis, obviously waves of activity traveling through a muscular tube. Obviously the neural activity is gonna be traveling waves also. The layout of our motor neurons in our spinal cord is such that our hip motor neurons are above our knee motor neurons, or above our ankle motor neurons, or above our foot motor neurons, right? And if you think of how you take a step, you flex your hip, flex your knee, flex your ankle, and then back in the reverse order. So a wave of activity traveling down the spinal cord will activate a locomotor-like pattern. And it's also the same for reach and grasp. And the innermost part of our spinal cord is the shoulder motor neurons, then the elbow motor neurons, then the wrist, then the hand. So reach and grasp, which is shoulder, elbow, wrist, hand, that EMG pattern literally has to be a wave of activity traveling through those motor neurons. Even the homunculus in the brain, we have sensory and motor homunculi, which are like little people in our brains. So if you're moving your leg, hip, knee, ankle, it's going to be, a wave of activity through that homunculus. I think the spinal cord is understudied. Cats can be retrained to walk and stand even with a completely severed spinal rat spinal cords can learn entirely new behaviors without the brain. And I like to think of movement as music played out on the piano of motor neurons. The motor neurons are somatotopically arranged, like a piano is tonotopically arranged. And it's like a self-playing piano, right? Because we can stand on our own. It's kind of like a background level of activity. And then, locomotion would just be like running your finger up and down the keys on that piano. The brain is the pianist. And I think we don't have a chance at understanding the pianist until we have a better understanding of the piano. And cognition and language have been proposed to be motor processing, which I find highly interesting, but highly speculative. And yeah, that will be it. This is the link to my to this presentation, so you can get to these links. There's some places that I found a lot of information that was very useful. You can click on those. And yeah, Rene Descartes said, "I think, therefore I am." I take the existence of the world for granted. I assume it exists. So I say, I eat, therefore I think. And thank you for your time. And I'm happy to have questions and discussions and all that now. I'm sorry.
Hi, can you hear me?
I can.
John QB. I've been looking for the hand raise icon, don't see it. So do other people see it? They can raise. So I have a couple of questions, lots and lots of questions, in fact, but a couple for now. One is, I have no difficulty with the standard view that the nervous system is an infolding of the ectoderm and is not derived directly from the gut. It seems to explain everything I need to know. The nervous, the CNS has the features of ectoderm. They're fine for me. Meninges are not part of that. They are underlying stuff. The folding, where it makes lots of sense. The anterior and posterior neuropore closing, zipping up from center, both, it all makes sense to me. So I don't see where the problem is there. The other, before I, we'll let you go. So this, it's almost all these ancestral developments are topological. So you stretch things, balloons, and I think that's great. But in my mind, that doesn't need to be the whole answer. When evolution occurs, it presumably there's underlying changes in the DNA, which are in all the cells of the body, not just a region. So the possibility of expression is not just topological. And I think there are examples of things, similar features with similar biochemical, cell biological features developing at different topological regions. So for example, myelin, I'm not really sure how that works out, but other sort of evolutionary inventions which don't follow beautifully this topological stretching thing, which is just a complexity. It doesn't really, that's just an observation. So the first thing is, what about the neural tube being well explained by an infolding of the ectoderm?
To me, it's not well explained, because the ectoderm, other than the central nervous system, is just the outside skin of the body and its basal membrane. Just as the endoderm is just the skin lining the gut.
So, yeah, I don't have a problem with that. But why do you say it's the gut? If the outside, the gut's already folded in and become a tube, and then secondarily a second tube formed, which is the nervous system.
Yeah. So I'm proposing that the gut was the original tube. The body tube is a copy of the gut tube wrapped around itself.
Well, there's an outer layer, which is ectoderm and endoderm embryologically. They're,
yeah.
I mean, no, then it infolds a little, and then they start calling it but they really seem like the same, almost the same.
I don't think ectoderm and endoderm are very, very separate, I believe.
gastrulation, they look like a continuous thing. There's someone drew a line to make a border. And then in my primitive sense of memory of embryology, that you have a ball first, and then it folds in.
Yep.
I'm a little, my memory is unclear. I don't have any difficulty with the similarities in the ectoderm and what classically called endoderm and ectoderm. They have a lot of cellular biological features that are really, really similar.
Yeah, so I don't, I think ectoderm, endoderm, and mesoderm should be replaced by tubes because
I'm not sure about mesoderm.
That's the skin. It should be an endotube, an ectotube, and then a bunch of mesotubes, in my thinking.
Okay, so let's get back to the first question. You have this. You already have the gut,
yep.
And then you have an infolding of the epithelial layer that forms the neural tube. It has nothing to do with the infolding of the gut. Why does that not work?
Well, hold on. Okay, so the earliest animals are just a gut. Their gut and body are one and the same. Single. The body. Then later in evolution, we are a tube inside a tube. Okay, and so you've got a gut tube inside a body tube, and then even later we get central nervous systems, which are this invagination of that body tube, and so I think the body is a first generation copy of the gut. And the central nervous system is a second generation copy of the gut. It's a copy of the body tube originally. An inverted copy of the gut. The central nervous system is what I believe it is.
Maybe we're just putting different words on the same thing.
Exactly. And this is, I have to sort of invent a lot of terminology here and stuff. So yeah, it's.
I mean, because your figure at the bottom left looks like two parallel tubes, which is the gut versus the nervous system, which makes sense. But
yeah, this exists in human embryos for a long time. So we have, we start with a hole in our head,
right?
And it dumps in. It's called the neuroenteric canal. Embryologically, it dumps into the gut, and then obviously both of those ends have to close off in order for us to develop properly.
Yeah, I've been not trying to incorporate the enteric nervous system yet, and maybe I'm wrong about that. I think the enteric is considered classically sort of a migration from the neural crest, which comes, is sort of, is it?
That is absolutely correct. In us, and this is an inconvenient fact, but I leave it out because I believe it's easily explained. In us, the neurons in our gut do not develop in situ. They do not develop in place. The neural crest develops a bunch of neurons which migrate to the gut and then fill it with neurons. But innervated guts existed from the very beginning of time. So obviously they originally developed in situ.
Here's a third question, maybe the second question. You use the term copy, the nervous system is a copy of the gut. And I was wondering what you meant by the word copy, because I can think of copying occurring a few ways in evolution or embryology.
Yeah, I think it's a literal copy. So imagine early worms that were just a single tube, like the Nemerdians, for example, just this single tube. Okay. Very quickly, you get predators, right? It's much easier to eat another worm, which is made of exactly what you're made of, than it is to sift tons and tons of earth looking for organic material. That takes a lot of time and energy. Much easier to eat someone who's already done that work for you. Okay. So you've got a strong selection pressure to increase in size. It will both prevent other animals from eating you and will allow you to eat other animals. And so I believe that this earliest tube, it may have been just a defensive mechanism to make themselves larger. Hey, wrap ourselves inside ourselves. Now we're twice as big. That guy can't eat us now because we won't fit in his mouth. Eventually that became permanent. And so originally, you got the enteric nervous system existing in the gut here. And now you've got the peripheral nervous system existing in the body here. Originally, they would have been exactly the same nervous system because it's just one tube wrapped around itself. It has to be the same nervous system. It's but then as soon as they're unique like this, they can start to specialize and differentiate to do different things.
Let me suggest I think different, maybe the same, but different idea, use of the word copy.
Sure. Yeah, I'm not sure that's the best terminology.
Yeah, in one stage in evolution, the genome develops a mechanism for making a tube called a gut out of the body, whatever the body is. And that's really good. So this mechanism has been developed. And this mechanism is potentially there in every cell in this primitive animal. So then something comes along that gives it the advantage of making a second tube. Well, it's got this mechanism anywhere. It doesn't have to be topologically. So it copies the mechanism in a different location and creates another tube then that becomes something else. That's one, what was going through my head when I heard the word copy.
So I had a follow-up question. Hold on, let me put my camera on so you can put a face to name. I'm Isaac. I'm a second-year graduate student, and I wanted to kind of bridge between what Dr. Kuby is asking and what a bunch of the people asked in the chat.
The chat, so please ask me questions from it.
Yeah, so and I think Salvador can kind of link it together after I ask this question, but on the note of the copy and on the note of what Dr. Kuby was saying, yes, you do see a lot of times throughout phylogeny that there's a gene product and then there's maybe a chromosomal duplication, and having two forms of that gene product allow you to actually get specificity and diversification of those two things. And I always like to refer to the red and the green cone in the eye. So do you predict experimentally that there might be some region of a genome that is the tube region that then had been copied and has evidence of evolutionary time mutations to it, like an evolutionary clock?
I think yes. So I like the gene duplication. We've apparently, the reason we got 23 chromosomes is multiple times in our evolution we've completely doubled our number of chromosomes. Just kept them all, and that allowed those genes to specialize in doing different things. In terms of these tubes, I think the most important thing genetically is the ability to form them. And so gastrulation is the original formation of that tube. And there are, I forget what they are, but there are certain genes involved with that. Deuterostomes, which are a big leap from protostomes, can pull in tubes wherever they want. So I'm guessing they use the same or a very similar genetic mechanism, perhaps a duplicated genetic mechanism to do that. So yeah, I would predict that pulling these tubes in the gut originally, the gastrulation, and in ourselves, the central nervous system and stuff, I'm guessing they would use the same genes, the same evolutionary origin of genes.
Okay, thank you.
Joe, both Bill and Erica had related questions. Maybe they can ask them now.
Yeah, sure. I can talk. Great talk, Joe. That was super duper interesting. Can you guys hear me?
Yeah, I can.
Okay, cool. So I think I asked sort of similar to what Bill kind of asked. So you make, I think, like a really great case for it feels like a sort of paradigm shift in how we look at all this. And so do you have any ideas in terms of, specific experimental or observation, sort of like specific next steps in terms of experiments or observations, or, like what could help provide evidence for this hypothesis in kind of a concrete, incremental way? Like, do you see anything like that that might exist?
I would like to release this idea to the world. I don't get paid to do this. And I don't have time. I don't have the time and energy to pursue this in the level of depth it should be pursued. So I don't know. I'd like to maybe form a team, a group of people who would all like to collaborate on this. And the first step is maybe publishing in a top-tier journal, trying to. After that, a book. This Joseph Ledoux, a friend just loaned me this. And this is a fantastic, big book of the evolutionary history of consciousness. And it's really, really good. It's almost the exact same story I just told, except it doesn't include the adaptable gut twist on things.
Uh-huh.
So, yeah, publish, book, video. Yeah, this is being recorded. So I hope nobody minds if I release that to the wild. I don't know what to do with it. Cool. Give me advice.
Okay, cool. And I also had kind of a second part of the question is, are there any areas kind of where you feel like this idea, like, are there any weak links that you can kind of see from afar? Like, are there any areas where you feel like this idea doesn't fully account for what we see in reality, or might need to be supplemented by something? Is there anything like that you kind of can see from a mile away?
there were multiple times when I was going through this. Darwin said, when he was developing the theory of evolution, he said, well, what you have to do is you have to try to prove it wrong. And so I've tried many times to prove it wrong. It actually you didn't even have to try that hard at first. A lot of things didn't seem to fit at first, like the breakdown between coelom theory and germ layer theory. I was like, man, this worm is acoelomate, so it's the lowest level of coelom, but it's a triploblast, so it's the highest level. The flatworms, for example, are the highest level of germ layer theory. And I'm like, that doesn't make any sense. This doesn't seem to work. But once you start seeing it as tubes instead of as germ layers, it works. What other problems? Yeah, the one problem which came up briefly earlier is that invertebrates, the neurons in our gut do not actually develop in our gut. They're part of the neural crest, which then migrates to the gut. I think that's pretty easily explained as embryological shortcuts to get there.
Okay.
Other weaknesses? I mean, yeah, not really.
Yeah, cool. Awesome. Well, thank you. It was a really, really good talk. I'm sold. Series of tubes. I like it. Thank you, Joe. That was really, really cool.
And I think Ivan has a question. He's been raising his hand for a while. You're muted, Ivan. You have to unmute.
Ivan, can you unmute?
Where is Ivan? I'll unmute him if I can find him. Oh, here. He may not know how to unmute. I muted him because he was muted. Before I'm trying to get him to unmute.
Dr. Kuby, if you're co-host, you might be able to unmute him by going into participants.
I went there and it says ask to unmute.
Oh.
I am muted.
Oh, I can hear you now.
I enjoy the straightforward plumbing presented.
Thank you. It's all plumbing.
Limbs move in not exactly tubes. Limbs have become very important all through.
Obviously, yeah.
But more recently, like in the last few decades, the lymphatic system of the brain has shown itself to be of some relevance to many of our functions. I was wondering if you had any thoughts of the relationship
Go ahead.
Of the twisted pipes
The
twisted pipes, and also the way our lymph is moved, both in the brain and in the periphery.
Okay, so I think that we got limbs. I'm going to show you a quick, a short video here. If people are interested, feel free to leave if you don't have time or whatever. But this is just a quick video. This was when I was first developing this idea. I thought, well, if I can draw this idea, I'll probably understand it a lot better. So I made this video. So the gut tube was first. It becomes specialized, obviously, after the body tube. You get lungs from there, your liver, pancreas, and gallbladder come from the gut. But obviously, you can't grow these things in space and drag them around. You have to have a place to put them. So where does the body tube come from? Or the bladder and the urogenital system also come from the gut. I think I tried to draw the heart, but I didn't understand it at that time, so ignore this bit. But it's about to get to limbs. So we grow all these internal organs. How do we keep them safe in space? We need a body to put them in. I think the body tube is the gut tube wrapped around itself. A second here. So yeah, I think the way we got to a body tube with an internal gut tube, a place to keep all the organs, is that body tube wrapped around inside itself. And then later, our pharynx is an invaginated bit of the ectotube, so we can have brain control of our pharynx. And then, yeah, so then the central nervous system is just the imagination of the body tube. Closes off at both ends. All the sensory apparatus is at the front, so you get a brain up there. This is the notochord, the stiffening rod that allowed us to become more fish-like in our movements. And this is where, yes, so now this is bones. This is, I think, where bones come from, is an invagination of that body tube that was originally an atrium. Gives you, so you this, you got ectoderm outside here. If you invaginate that into yourself, you've got two layers of ectoderm right next to each other, which can produce an exoskeleton, right where we see our ribs and our vertebrae and stuff. So so once you've got these primitive bones starting to form, then and yeah, we also have the external control of our anus, which is very useful. And so, yeah, you can just, you can pop limbs out of this, in my impression. Hold on a sec. Yeah, we have a postanal tail, which is important. The vertebrae formed. Let's go back to see the limb. So yeah, so basically, once you've got some bones inside you, it's fairly easy evolutionarily. I can make new bones. We already know how to do it. So yeah, originally, we're just like a tube. But it's nice to have limbs. It's nice to be able to move through the water and stuff. And so I think that's just a matter of, these bones being used to push outwards into the body tube to make external limbs. And I hope that clarifies what I think you were asking.
Okay, so we've gone on a little bit. Salva, do you see questions? My view is not that great, but I want to look forward to
So I think Sid had a question and also Isaac, but yeah, I don't know if we need
Well,
to.
Sid and Isaac are graduate students, and there's going to be a meeting with the graduate students afterward, so maybe they can wait.
Sounds good.
Yeah, that works.
Okay, so yeah, so what we've been doing in the last few weeks is everyone leaves except the speaker and the graduate students have their lunch virtually. Once we should stop the common meeting. Does that work?
Yeah.
Yeah, that works for me.
Okay, so I'll invite all the non-graduate students to leave and the graduate students and Joseph to stay. Oh, just a general comment that what he's trying to do is incredibly hard. The history of looking at the nervous system from an evolutionary perspective is old, but it's diminished a lot. And
fortunately so.
Great project, and one that should be renewed.
Well, thank you all very much for your time then.
Thank you.
Thank you so much, Joe. Great talk.
Thank you so much.
Thank you, everyone. Thank you, Joe. That was fascinating. I'm going to stop the recording now.
Okay, perfect.